And so then one thing I also added for day 3 is something we discussed for using multiple species in the distance sampling. So if you scroll down to the very end of this HTML, after the exercises, I added a bonus section when I used some life history traits and used those in distance sampling. And then I compared a model where we have just species as main effects. So this is like almost like estimating them separately. You can see estimates vary quite a bit. And then I used body mass, maximum singing frequency, migratory status, habitat preference as predictors instead of the species themselves. And then it turns out this model with fewer parameters and some traits is much better supported. So I stacked the data and then used the same cmulti function. And then you can see here that for small sample sizes, when I'm not using a predictor, just species as fixed effects, I get all these differences between the two. So when you have more sample size, the two tend to agree. When you have smaller sample sizes, you get more extremes when you are not smoothing it over with some kind of life history predictor. So what you can do on your own, if you have time and you're into this multi-species stuff, is you can do some kind of leave one out cross-validation. So use all the rest of the species, estimate constant effective detection radius. And for that species, just take all the data and estimate it separately and see how well you can predict based on body size and related things. And that way you'll get a sense of how well this is working. So if you happen to have a species without any kind of data in the same region, would this be still helpful to come up with a tau estimate, even though you don't have the kind of breakdown in multiple distances that you could use. And we are going to see what kind of data you might have where this is the case, that you don't have the distance information, but you still would like to do something for a species, and what can you do there. Another thing you can do is, so this only looks at a constant tau and how to estimate that using something like x1, x2, x3. So what if, as you recall, this was a function of land cover type? So if you want to incorporate land cover type slash species traits, then you would need to have, for example, habitat class as a fixed effect, and you would need interactions between habitat type and these life history traits. So that way, the fixed effects for the life history traits would contribute to the intercept for each species, and then the interactions and the main effect for habitat is going to be like the slope. So I'm not going to go into the details. I might add this later because I think it's a pretty neat extension, and I like the way how it turned out to be, even without any kind of phylogenetic regression, straightforward and quite meaningful results. But just if you want to take it from here, then you can also do that. So this is all what I wanted to share regarding yesterday. And then let's begin with a few slides. So you can review how this all comes together. Yes?
Oh, yeah, before you continue, I need to ask a question about that. So sure. In this case, if you're adding species level information, like traits, and even if you have a fixed effect of species, don't you need a random effect of species, not only for the accounting for dependency, because you have many observations from cluster among species, but if you want to generalize to species not sample, or pull information from other species to some that you don't have detections, wouldn't like the random effect structure help?
You can use random effects. It is not going to help you a lot in predicting your like expected value for tau, it is going to capture the
Count
error
from-
on that estimate probably better.
Yeah.
Because then you would need to incorporate that error, which is the distribution of the random effects, so that that next species is coming from that distribution. So it might not be exactly this number what like body size would predict, but it might be in this range because there is this other variation which is not explained by any of this. So kind of getting an idea about that range of possibilities is where the random effect would be helpful. And then that's where the phylogeny might come in, that would help you reduce this uncertainty because it just adds more information.
Thanks.
All right. So last time we ended up with putting it all together, but we haven't really done a lot of analysis. And we also haven't checked that at day 4, we were looking at regression techniques and kind of learned how the different methods might lead to very different conclusions about the mean abundance. And we tried a bunch of different techniques to adjust them, but nothing really worked out great. And that's why we then spent 2 days of learning about the time and the distance component and how to use that information to account for at least the methodological differences. So today we are going to see that this is really working. And then the only thing that remains is how do we interpret these estimates? Is this really abundance, or do we still need to think hard about the assumptions and whether this is still just relative abundance? And once we get finished with that, then we're going to talk about how to incorporate human-based point counts with recordings that are done in the field and then analyzed later, and also how to deal with roadside surveys. This is the area where I think the research is ongoing, and I can just easily summarize. It's like, oh, you can do this, you can do that, or a combination of both, and then you'll see. So I'm going to less likely to give you like the definite answer. Well, I would do A, but I might say, well, let's try 3 different options and see which works best. All right. So again, go to the releases, download, say, QPAD local day 4 so that you have your own copies, although there's not going to be day 5. So unless you update your GitHub project, you won't see any new changes from me. So just having a hard time seeing this title because of the Zoom. Okay. So these nuisance variables that we spent 2 days learning about the P and Q, now we understand how we can estimate these quantities based on removal modeling and distance sampling, and how this relates to when individuals are present, then they need to be available for sampling, which means they need to show some kind of behavior, which then we can further detect. And then the detection process depends on the distance and the physical aspect of this act, and then also how we perceive that distance, and possibly multiple species interacting with each other and the observer, and then just that mental process of keeping track of multiple things. That might be another deviation from this idealized condition that we handled so far versus the real data. And so just to go back, what QPAD means, we have the expected value of our counts, which is what we observe, 0 or 2 Tennessee warblers. And then we think that's a combination of the true abundance. There were 5, but we really observed 2, and some kind of correction. So if you can figure out this correction, then we can go from 2 to 5. And that way we would know what n is. And then also n can be written as the density per unit area times the area. And then we also then said C is a function of P and Q, which is the availability part and the perceptibility part of the detection. So then if we reorganize, we get QPAD, and then rearranging this equation, our estimator for density is the mean count divided by A times C, which is this area times our P and Q estimates, which we saw how to calculate. So this is the main idea, and we are going to see how we can extend this further. So we are not just talking about the mean count here, but how to do this in a regression context when we want to know, like at each location, what is the expected abundance as a function of land cover, climate, and so on. So what we were doing in regression, how we can at each sampling station or each visit use that information to come up with service specific P and Q values and to use it in a correction factor or as a statistical offset. And we are going to see many different examples for that, which I think is going to be a good starting point for any of you who would like to do this kind of analysis. So it's more like a recipe. If you want negative binomial regression, go there, grab those few lines, and that's how you fit the model and do the prediction with the offset involved. And we've covered how to calculate those offsets, but there's also going to be just a recipe recap. Here is how you do it. So now we are going to focus more on these applied aspects. Now you have the data and you know all about P and Q and what those mean and what kind of assumptions come into the picture. And now you're only interested in getting some results and then look at them and think about them more. So in our 2013 paper where we published this QPAD methodology, then we looked at how the different distance information and time information, if you just combine the counts and get the mean density, and each dot here is a species. We had, I believe, 90 some species altogether in the Canadian boreal. Now you can see if you just use subsets of the same data set, I mean subset that we have these multiple bands, and let's just use 5 minute 50 meter, 10 minute 50 meter, 5 minute 100 meter, 10 minute 100 meter, then you can see your estimates are going to vary quite a bit. With time, less so, but with increasing area, you can see going from 50 to 100 meter, you see these mean densities at least double. Now, if I only take into account that I know the area, so I can use the log of area as an offset, and we'll see how the correction relates to the offset, but you can now guess it's a log function. If I only use area, I get them closer together. So less variation, but still there's this time component, and now it goes the other way. So if I have 100 meter, then kind of smaller, but it's much better. So it's better than nothing. If you want a quick fix and you don't want or don't have other information, you might just go with the known area and get much better. So this just highlights if you were to put together data sets with varying methodologies, then you could use the log area and possibly something about the protocol as a fixed effect to get around this. Even better if you can estimate some removal sampling parameters. Now you see that these two bars are kind of similar in height. That means we could correct for the time variation, a time-related variation in the counts. Same thing goes if you use this log area times Q, which is now the distance-related one. So now this first and the third are kind of similar in their mean, and the second and the fourth. So now we adjusted for the area difference quite well, but still there is this difference due to the time, 5 minutes versus 10 minutes. So if you take into account both of those sources and the known area, which is our A times P times Q quantity on the log scale added as an offset, then you see there's variation because there are species with less data, and there we would get something else. But on average, across all these 90-some species, we see that the mean of this distribution is very close, which means on average, we were able to adjust for methodological differences. And so whether that number and its relative mean density, because I just decided this is going to be the reference, this is where we have highest number of counts and we are using all the information we have, and let's just compare relative to that what we get. So whether this abundance is a true abundance, so we can say it's 2,562,822 bird individuals in a region, or that's not quite correct. And here's an ovenbird example, and these bands kind of show you the different versions of the corrections. So on top, this is when you have all the information. So it's kind of the same idea, A times P, A times Q, A times P times Q. And what you see here as part of this shading is the uncertainty around the parameters of this ovenbird density. So the gray area is coming from the GLM that relates to abundance. And then this white area in between relates to the uncertainty that is coming from the offsets. So as we estimated the offset, those intercept and slope and whatever parameters related to phi and tau are also random quantities which we estimate and come with some standard error variance covariance structure. So if you take that into account, then you can see it's much smaller compared to what uncertainty we get around the density related things. And then here you can just see how abundance varies across the different land cover types, dominant tree cover, and you can kind of see that the relative patterns are very similar as we go from one to the next. So these are ranked in decreasing order, and also how the offsets are modifying these are also the same. But one we like to think is the true abundance, and the rest are just versions of relative abundance. So this is how you can kind of think about the uncertainty around these. And we are going to do an example how to capture these uncertainties in the same model so that you can have this uncertainty that represents both, or actually all three of them, because for P and Q, there might be some uncertainty, and then for density as well. So when you integrate different projects that are coming from various protocols where the total time area you sampled varies, then you want to normalize these. So if you take the projects, then you can identify a subset where you can fit removal models, which requires at least 2 time intervals at the same survey. So 0 to 3, 3 to 5, or something like that, or maybe more than 2. So if you have something like that as a subset, then you can take those and use the C multifunction, or I'm pretty sure there are other packages out there, and get this phi estimate. Then move on to step 3, identify multiple distance subsets of the data. So you can imagine that some project might decide, let's do unlimited distance count without putting individuals into distance bins, but do 5 time intervals, 1, 2, 3, 4, 5 minutes. The other might say, well, let's just do 5 minutes and 2 distance bands, 0 to 50, 50 to 100, or 100 and above 100. And once you have those, then there's going to be these different subsets that you can use to fit the removal model and the distance sampling model, which would then give you a way to predict P and Q based on the total time or the distance interval or various factors that went into those models, for example, time or date of the survey or land cover type, which, by the way, is also available for all of your data points. So that gives you a way to predict those for surveys that you didn't use to make those estimates, right? So now you take this subset and make a prediction for all of the data set, so all the points. Because you might have something like BBS, 3-minute unlimited. You won't be able to include it into the removal modeling or in the distance sampling, but you know when the survey happened, on which day, and maybe what the land cover type was, if you know the location, or you took note in the field, and that way you can make a prediction regarding P and Q. So in step 4, you make these predictions for all the surveys, and then calculate this offset, which is the log of A times P times Q, or for unlimited counts, for each of those points, you just take the effective area times P, and we saw why that's happening, so how you can replace Q with an effective area. And once you do this, then it's really up to just going through the usual steps of modeling, finding which predictors best capture the species abundance and how that varies through space and time, and what kind of error distribution to put around the counts. Is it Poisson or random effects, or how to fit those using GLM or machine learning techniques or even MCMC. So these are decisions you need to make, not based on this workshop, but based on what people otherwise would recommend for a given system, species, or what you are familiar with. So these methods can deal with multiple methodologies, but then interpreting these results still requires some just articulating caveats, limitations, and that is going to revolve around the assumptions and how likely those are going to match, whether you can assume that these results at the end are still going to be the real D, which is density. So if you can't estimate P and Q, let's say this is the case for a species, this can happen because you don't have that species information in any of the projects where you have this multiple time and distance interval. So then you can look for similar species. And this is what we discussed before. And you can check out some of this in the LH-Reg package documentation, what we did for that ecography paper and what I just showed you regarding the previous day. Look at the phylogenetic relatedness, collect some trait information, and try to pull the species data that are very similar and get an estimate, or substitute closely related species information for this species as an offset. And that's why you can do some kind of validation to make sure that this is going to be a right approach before actually doing it, but this is doable. Another way how you can get this offset if you don't happen to have the data, to see if you can use estimates from similar studies. So you don't expect body size and related physical aspects of sound transmission change across 500 kilometers or 2000 kilometers. Physics and species biology is roughly the same within the range of the species. And if you are in similar habitats, you might expect similar distance relationships. So you can rely on other studies. And this is something I've done in the past. We looked at the data and it was really small, and small in the sense that 200 data points takes a lot of time to collect. But still, if you don't have enough detections for a species, you can't really do much, even though you had multiple time intervals. Those estimates are just going to be really uncertain. So we decided to just take estimates from much larger study, the Boreal Avian Modeling Project. We have published these offsets. And if you follow this link, you're going to see a little project which describes how you can extract information and generate offsets based on our latest estimates. And we are in the process of updating these numbers based on more data. So basically, you need to know what species you are interested in and whether we have estimates for those species, date, time, let long, and the maximum duration and distance, because this is just going to influence that cumulative function that we are using for the offsets. And once you know that, then you're just going to get an offset value that you can work with. So if you are really running out of options and your data set is small, or for a particular rare species you don't really have an estimate, you can look for similar species or other studies where, based on much larger data set, you're likely to get a reasonable estimate that you can use. So some of the advantages that I could think of for this approach of standardizing different data sets, and also the methodology that we have used, so we estimate these components independently, is you get a smaller parameter space. And I'm going to show you an image which would make these claims clearer. And this smaller parameter space and the fact that we are doing this in steps and not jointly in a single model makes model selection more straightforward, because you can concentrate on one aspect of the system and just get done with it, move on to the next level. Less collinearity, which means you can use the same predictor for distance sampling and for density, because land cover is going to influence the distance function and also going to influence the abundance of the species. So you can use the same variable. It is not going to lead to any kind of confounding. And also it's quicker because we can only take subsets of the data where we have this multiple band or time interval information and some detections. And that is just going to make it much faster to get estimates. So this is what I was talking about. We are looking at three parts of a mixture model in a way, where we have this true density that we are really interested in, but we also have to estimate P and Q related to removal and distance sampling. So you can write a joint model, for example, in JAGS or STAN using some MCMC method, which makes it really easy to put all of these together and just do joint modeling, in which case you can look at all the possible combinations. For example, if you want to do model selection, then you can look at X1 in this part of the model, X1 in this part of the model, and then so all of these combinations might come into play. You need to assess, is this a better model than this one? And you might have the same covariate for density, removal, distance, but some of those might be unique for each of these. So instead of that, in our conditional likelihood framework, we can say, give me a subset, deal with removal sampling. Give me a subset, deal with distance sampling. Once I know these, then I can plug them into my density model and I can focus on these. So the smaller parameter space refers to, instead of 36 options here, you're looking at only 12. Which, if you need to make all of these combinations and compare them and come up with a final answer, then you can do them in steps. And that just saves time and simplifies everything. There are also disadvantages. I had another one listed here and I didn't update the PDF, but that's basically taking care of uncertainty needs more work, because then you need to propagate these uncertainties into this model, and that is something often you have to do by hand. In a joint modeling, that is taken care of. And the second thing which is missing here, also model validation. When you want to do some goodness of fit testing or some kind of AUC type metrics to calculate, that is going to require you to be very careful about what your predictions are going to look like. And we are going to look into that, what I mean by that. So the expected value is the true density plus all the offsets, right? And when we want to talk about density, we don't want the offsets. When we want to compare our predictions to the observations, we need the offsets. So that is just something you need to keep in mind when you want to do some kind of validation goodness of fit, and you're interested in the expected value that leads to the counts, then you need to incorporate the offsets, which is again an extra step to make sure it's there. Otherwise, you would get really surprising results.